Imagine two sounds with the same number of pulses. A mouse should treat them the same, but it keeps responding differently, and other mice make the same kinds of mistakes.
The same mistake pattern across animals. That sounds less like random noise and more like the brain is nudging the decision.
Exactly. Today's paper asks whether the shape of a sound map inside the auditory cortex can explain why perception gets biased.
First, what do you mean by a sound map in the brain?
When a sound reaches the ear, the brain converts it into patterns of activity across neurons. A neuron is a nerve cell, and a pattern means which cells respond strongly, weakly, or not at all.
So each sound leaves a kind of fingerprint in neural activity.
Yes, but the deeper idea is that the brain does not store those fingerprints as isolated dots. It arranges many sounds by similarity, so sounds that evoke similar activity patterns sit near each other in a representational map.
Wait, so it is kind of like a city map, except distance means these sounds feel similar to the brain.
Great analogy. In neuroscience, representational geometry means the shape of that map, which sounds are close, which are far apart, and which features form the main directions through the map.
And the paper says that geometry can bias decisions.
That is the key question. Traditional decision experiments often focus on a small set of task sounds, asking whether an animal can tell A from B. This paper asks a wider question: where do those task sounds sit inside the larger sound map?
So instead of asking only whether the mouse learned the rule, they ask whether the whole map makes some rule following harder.
Exactly. The authors studied pulse sounds, which are 1-second sequences of brief white noise bursts. White noise is a hiss containing many frequencies, so here the important difference between sounds was timing, how many bursts occurred, and how they were arranged.
Timing is very Oskipod. It is rhythm, not melody.
Right, the sounds differed in pulses per second, from 3 to 11, and also in the detailed temporal pattern. Because their spectral content was largely similar, the brain had to integrate the timing structure.
Where does the bias appear in the behavior?
The mice learned a go/no-go task. In a go/no-go task, one kind of stimulus tells the animal to make a response, and another tells it to withhold that response.
What was the rule here?
Sounds with 8 to 11 pulses per second were go cues. If the mouse poked, it got water. Sounds with 3 to 6 pulses per second were no-go cues, where poking led to a mild air puff. 7 pulses per second was an off-target middle case, with neither reward nor punishment.
So the reinforced feature was just the number of pulses per second.
Yes, the researchers trained 11 mice on 90 pulse sounds, and the animals learned the category rule over about 1,000 to 2,000 trials.
Then if two sounds both have, say, 6 pulses per second, the mouse should treat them similarly.
That would be optimal under the rule, but the mice showed substantial differences among sounds with the same pulse number. Some sounds pulled the animals toward poking more often, others pulled them away.
Could that just be individual quirks?
The striking point is that it was shared across mice. The authors normalized each animal's responses within each pulse category and found coherent bias patterns across individuals, especially for intermediate pulse numbers.
So the paper needs a mechanism for why irrelevant timing details influence a pulse counting decision.
Exactly. The central concept is representational entanglement. That means different stimulus features are not cleanly separated in the neural map. A feature that matters for the task and features that do not matter can point in partly overlapping directions.
Like a compass needle getting tugged by a nearby magnet.
Sharp, the mouse is supposed to follow the pulse count axis, but other timing features can pull the neural representation toward the go side or the no-go side.
What were those other features?
The key non-task features included pulse imbalance, first pulse interval, and irregularity of accents. Pulse imbalance describes whether pulses are more concentrated earlier or later in the sound. First pulse interval is the time between the first two pulses. Irregularity of accents describes how unevenly clustered close pulse events are across the sound.
How did they connect mouse behavior to the brain map?
They used three main steps. First was the behavioral task we just described. Second was an auditory cortex lesion experiment. Third was large-scale calcium imaging from the auditory cortex.
Auditory cortex means the part of the cerebral cortex that processes sound, right?
Correct, it is a sound processing region on the surface of the brain. To test whether it mattered for the task, the authors trained another group of 18 mice on a simpler discrimination: 3 pulses per second versus 11 pulses per second.
Then they damaged the auditory cortex.
In 9 mice, they made bilateral lesions, meaning they ablated auditory cortex on both sides of the brain. The other 9 mice had sham treatment, where the surgery was performed without cortical damage.
What did that show?
1 day after the lesion, discrimination performance dropped by about 20%, while the number of trials the mice initiated did not significantly drop. That suggests the deficit was not simply because they stopped engaging with the task.
But the Japanese script mentioned recovery.
Yes, performance largely recovered over 3 days, so the authors interpret auditory cortex as contributing to the task, while other cortical or subcortical circuits may also help or compensate.
Now the imaging part. What is calcium imaging?
Calcium imaging is a method for watching neural activity indirectly. When neurons become active, calcium levels change, and fluorescent indicators can make that activity visible as changing light.
And two-photon imaging.
Two-photon imaging is a microscope technique that lets researchers record activity from cells inside living brain tissue with relatively fine spatial resolution. Here, the team recorded layer 2-3 neurons in awake, head-fixed mice.
Were these the same mice that did the behavior task?
No, the imaging used an independent group of 10 behaviorally naive mice. They listened passively to all 189 pulse sounds, while the researchers recorded from 33 fields of view, about 3 fields per mouse.
So they measured the map in one set of mice and behavior in another set.
Exactly. For each sound, they built a population response vector, meaning a list of activity values across many neurons. Then they compared every pair of sounds using representational similarity analysis, a method that asks how similar two population activity patterns are.
That gives the sound map.
Yes, the result was a 189 by 189 similarity matrix, every sound compared with every other sound.
What did the map reveal first?
The auditory cortex represented more than simple pulse number. Neuronal population responses differed not only between sounds with different numbers of pulses, but also between sounds with the same pulse number and different temporal patterns.
So the brain was listening to the rhythm details, even when the task rule only cared about count.
Exactly. The authors described every sound using 11 interpretable features, covering overall energy, pulse regularity, and how pulse density changed over time.
11 features sounds broad. Did all of them matter equally?
No, multiple regression, a statistical method for asking which variables best explain a pattern, pointed to 4 major features in the cortical representation: pulses per second, pulse imbalance, first pulse interval, and irregularity of accents.
The task feature plus 3 distractors.
Right, distractor here does not mean the sound had extra noise. It means a feature that was not reinforced by the task rule, but was still encoded in the neural map.
And the axes were entangled.
Yes, the feature axes were moderately correlated with each other. In plain terms, the map did not place pulse count, early-late imbalance, first interval, and accent irregularity at perfectly independent angles.
Then they built that distraction index.
They did. The distraction index summarized how much a sound's neural representation projected onto the 3 non-reinforced feature axes, weighted by how aligned those axes were with the pulse count axis.
So a high distraction index means the irrelevant timing details are pulling in a direction that looks decision relevant.
Exactly, and that index predicted the behavioral bias across sounds. Across 85 valid task sounds, the correlation between distraction index and normalized poking behavior was about 0.32.
That is not the whole behavior, but it is meaningful because the imaging and behavior came from different mice.
Precisely, the explained variance was modest, around 10%, but the cross-animal prediction is important. It suggests that shared sensory map structure can account for shared decision biases.
Were there other behavior links?
Yes, the researchers compared decision strategies using distances in the neural map. Poking probability was strongly related to similarity to the reinforced sounds, with a correlation around 0.66, while similarity to punished sounds alone was weak.
So reward sounds acted like an anchor.
That is a good way to put it. The behavioral gradient looked more like how close is this sound to rewarded examples than how close is it to punished examples.
Why would the auditory cortex entangle these features in the first place?
The authors suggest statistical learning as a likely explanation. Statistical learning means the brain absorbs regularities from experience, such as which sound features tend to occur together.
For example, more pulses in 1 second might naturally go with shorter intervals between early pulses.
Exactly, if features co-occur in the sound world, a sensory map may encode them in related directions. That can be efficient, but it also means one feature can influence judgments about another.
So the bias is not just a failure, it is a side effect of a useful map.
That is the nuanced point. A map that captures relational structure can support perception and learning, but when the task demands a clean separation, entanglement can produce systematic errors.
And this fits the OSCAI pod theme because timing patterns become neural geometry. Then geometry becomes decision bias.
Well said. The paper ties temporal structure, population activity, and perceptual choice into one chain.
What are the limits of the study?
One limit is that the lesion effect was transient. The authors note that acute reversible manipulations and control region lesions would be needed to define the specific role of auditory cortex more precisely.
And the imaging had limited timing resolution, right?
Yes, the calcium recordings were taken at 5 Hz, so they cannot capture fast electrical timing directly. The authors focused mainly on spatial population patterns integrated over the sound and offset period.
Also, predicting around 10% of the behavioral variance means most of the bias is still unexplained.
Correct, the study does not claim a complete account of decision-making. It shows that representational geometry in auditory cortex explains a significant, shared component of perceptual bias.
So the story is that mice were trained to count pulses, but their brains also represented other timing features, and those features bent the decision.
If I had to put today's surprise in one sentence, the bias was not just in the mouse's choice, it was already in the geometry of the sound map.
The map itself made some sounds feel closer to the rewarded side.
Exactly. Saylor, Casetta, Noda, and Rumpel showed that auditory cortical representations of pulsed sounds contain entangled feature axes, and that this entanglement predicts shared perceptual biases in mice.
It makes a simple decision feel less simple. The brain is not just reading a number of pulses, it is reading a whole rhythmic landscape.
Today's paper was Representational Geometry of Sounds in the Auditory Cortex Explains Biases in Perceptual Decision-Making in Mice, published in Cell Reports in 2026. Thank you for listening to OSCAI Pod.
Thank you, see you next time.